We already know a lot about enzymes, right? Let's talk a little bit more, and we're going to be talking about what makes enzymes work more quickly or what makes them work more slowly. So before we do that, let's talk a little bit about naming of the enzymes. I already told you that it is currently the convention that molecules, which are enzymes, should have names that end with the letters ASE. Right here, ASE. So lactase or polymerase or ribonuclease or any of those things are going to be enzymes. When it comes to the sciences, you can trust that. And then the rest of the name is supposed to give you a hint as to what that particular enzyme does. So phosphatases, they remove phosphate groups, and synthetases are a part of dehydration synthesis. DNA polymerase makes polymers of DNA, all of that. Now, this next slide, don't worry about knowing the names of any of these. I just thought you might find it interesting that when you've got a blood test, like, the doctor says, oh, well, let's check your blood to see. Yes, they look at the cells in your blood, but the other tests that they run, a lot of those, they are looking at enzymes that are found in your bloodstream. So alkaline phosphatase, oh, there's a phosphate, an enzyme that adds a phosphate. What kind? Alkaline. Oh, that's a hint to something. And this particular enzyme is very abundant, particularly in liver and in bone. So if we find too much of it in the blood, we think, is something wrong with the liver? Is something wrong with the bone? Right? Amylase, this is an enzyme that is very common in the pancreas. So if we have too much amylase in the blood, we think what's going on with the pancreas. So when you do a blood test, those are enzymes. And now when you read them and your dad says, whoa, alkaline phosphatase is elevated, you can say, that's an enzyme. What makes enzymes work better or work worse? Well, first of all, enzyme activity. We will measure the activity of an enzyme either by how quickly the substrate goes away or how quickly the product builds up, right? So remember, we're thinking of enzymes as like robots. And if you put a robot into a room with its substrate and you wanted to measure how quickly does that robot work, you can either measure how quickly does the substrate go away as he takes it and rips it apart, or how quickly does the product build up, right? Now, in the labs that you will be doing, the virtual labs you will be doing with this lecture, we will be measuring how quickly one of the products build up. Which of the products? Well, which are the two products? The two products are oxygen and water. And we are going to measure how quickly the oxygen builds up by measuring how much bubbles get made. Okay, so how quickly enzymes work is influenced by the temperature, by the pH, also by how much enzyme there is, and by how much substrate there is. Yeah, I think those are the three things that we're going to be talking about. There are other things that influence how quickly enzymes work, things like cofactors and coenzymes, and there are other things in the environment that can interact with that robot to make that robot slow down or speed up. But we're going to be focusing for this lab on temperature, pH, and enzyme. And I need to tell you that when you do the enzyme labs, and when you are getting the results of those labs, you should write them down, particularly when you get those graphs that have the pH doing this, right? I want you to write those down so that when you take your quizzes, you will have something to go back on. At least take notes, all righty? Now, enzyme activity, temperature. Enzymes work more quickly as they warm up until they get to an optimal point. And when you heat them past that little optimal point of action, then the enzyme will start to be damaged by the excess temperature, and the enzyme will be denatured. You need to know that word, okay? Denatured. We will say that a protein has been denatured if something has changed its shape. So remember, proteins have got shape, primary, secondary, tertiary, and they've got an active site. As you heat up an enzyme, it's like a robot that goes from really cold, so he's kind of stiff. Oh, he's so cold, he's so stiff. So he works slowly. Then as you warm him up, he works better and better and better and better up until the point that he's so loosey-goosey that he starts falling apart. And as he falls apart, he has become denatured. Write this down. When a protein is denatured, it means that the protein's shape has been changed, and excess temperature will denature proteins, particularly enzymes. Okay? This changes their shape and destroys their function. Great. Let's have a look at that. Oh, this is not a protein. This is a simpler molecule than a protein. But it reminds me to explain to you that when we're thinking about molecules in general, it's not quite as simple as I said at first. At first I said, oh, they've got a shape, and that shape is as tangible and three-dimensional as your chair. Well, that is true. They're as tangible and three-dimensional as your chair, but they're not very good at holding still like your chair is. All molecules, they move a little bit. That molecular world is just a very jittery environment. So if we were taking a picture of this molecule, whatever it is, if I turned down the temperature, and if it went from human body temperature, which is 37 degrees, and it got colder, then the atoms would jiggle less and less and less and less. And when we got to 0 degrees Kelvin, they would hold still, right? And then as I warm them up, they would jitter more and more and more and more. And then with this molecule, if I boiled it, it might be jittering so much that it kind of flies apart, and then it would denature. All right? So that's the effect of temperature. If I were to graph the effect of temperature, and by the way, in these online labs, you will be graphing the effect of temperature, you will see that enzyme activity will be very slow, not necessarily zero, but slow as we get close to 0 degrees centigrade. And then as you warm them up, they get faster and faster and faster and faster. Now, 37 degrees, that's human body temperature. And actually, human enzymes will keep working a little bit better after you get to 40 degrees. But when you get past 40 degrees, they will begin to denature. And as they denature, they will quit working well. And you may know that every year, dozens and dozens of children die by being left in hot cars. And one of the reasons that they die is because as the temperature goes above 40 degrees centigrade, so that's, I don't know what that is in Fahrenheit, but as it goes above 40 degrees centigrade, their enzymes start denaturing. When your enzymes don't do their job, then your body can't stay alive. Right? One thing, denatured enzymes do not come back from that. Okay? I can take proteins and I can cool them off and warm them up and cool them off and warm them up. And when I cool them off, they work more slowly. When I warm them up, they work better. But when I go to the point of denaturing them by temperature, they don't come back from that. So that's temperature. What about pH? pH is a little bit different. With pH, there is an optimum pH for enzymes. And then as you go to alkaline or to acid in your pH, then you will denature the enzyme. So enzymes are very finicky when it comes to the pH that they like. And the enzyme, that protein, will be working at its optimal at that pH. Now, as humans, we tend to think, oh, a pH of 7, that's perfect. That's the one everyone likes, right? No. Okay. It's true that the enzymes that are most abundant in life are found inside of our cells. And inside of our cells, the pH is just a little bit more alkaline than 7 for humans. And so, yes, for most human enzymes, they kind of like a pH of 7, 7.4. But there are many enzymes that we use to help us digest our food. And those work outside of the cells. And you will find that this enzyme called salivary amylase that works in your saliva, it does like a pH of 7. But this enzyme called trypsin, it works in your small intestine where the pH is closer to 9. So it likes a pH of 9. And if you put it at a pH of 7, it doesn't work very well. Then there's this enzyme called pepsin. And this enzyme called pepsin, it likes a pH of 2. Are you crazy? That is almost straight hydrochloric acid. Yes, but pepsin is not particularly tough because if you bring it to a pH of 7, it won't work at all. All right? By the way, I want you to notice that pepsin and trypsin, their names don't end in A's. What is up with that? Well, they were just named really a long time ago. So they actually do have modern names, but no one uses them because we've been using the names pepsin and trypsin for so long. That's the effects of pH. What about the effects of substrate concentration? Well, let's think again about our robot. If we had our robot who was supposed to rip apart hydrogen peroxide, and in our room that we put the robot in, we put in hydrogen peroxide, but just like 10 of them and they're kind of hidden, then we would not expect him to be able to rip them apart very quickly. So we would not expect our enzyme to make oxygen very quickly in that environment. But if we put our robot into a room where there was just hydrogen peroxide all over the place, so as soon as he ripped one apart, there was one right there, he could work very, very, very quickly. Now, when there is much substrate around that the enzyme is working as quickly as he knows how, like grab, rip apart, boom, grab, rip apart, boom, that's as quickly as he can work. If we add even more substrate after that, then we will not get the robot to work more quickly. He's already working as fast as he can. So when you look at a graph of the effects of substrate concentration, it doesn't go up and down. With pH and with temperature, it goes up and then down. But with substrate concentration, the enzyme will work more and more and more quickly as you give it more substrate until the point that he's like, okay, I'm working as fast as I can. It doesn't matter if you bring me more substrate. This is as fast as the robot that I am can work. So with the effects of substrate, it'll go up and then it'll plateau. It doesn't go back down again. Last thing I need to tell you is about coenzymes. Many of our enzymes in the human body they're not complete by themselves. Here is the protein, and the protein has got an active site, but it's got like this little chunk there that needs to be filled by a coenzyme so that it makes a perfect active site. Those little helper pieces are one type of those helper pieces are coenzymes. And for this lab, you need to know for the quiz that coenzymes, they are organic molecules, but they are not proteins. They are not enzymes themselves, but they are made from water-soluble vitamins. So vitamin B, all of the B vitamins and vitamin C, they are water-soluble vitamins. And the reason that our bodies need those enzymes, you know, your mom says, take your vitamins today, right? or you should eat your vegetables because they're full of enzymes. I'm sorry, they're full of vitamins. The reason we need those vitamins is because they are where our body can get coenzymes, and the coenzymes help our enzymes work properly. So without vitamins, your enzymes cannot do the jobs that they need. Like B vitamins allow us to make healthy skin and hair. Why? Because B vitamins, particularly, I think it's vitamin B12, I don't know the vitamin, but it's where we get this little coenzyme. Without that little coenzyme, the enzyme's not good at doing its job. But with that little coenzyme made from the vitamin, now it's really good. The enzyme can be good at doing its job. That's it. I'm going to also post a video from Crash Course. Make sure you do a good job on all of your enzyme labs through Connect. By the way, if you find, no, I'll do that on a different video, okay?